Good morning, friends. So, in the last lecture, we had discussed about the structural chromosomal aberration. Now, another part of that lecture is the numerical chromosomal aberration. So, let us see what is numerical chromosomal aberration. So, in last class, we have already discussed that chromosomal aberration is of two types. The first one is structural aberration, and second one is numerical aberration. So, in numerical chromosomal aberration, this has been divided mainly into two parts, that is euploidy and aneuploidy. So, let us start today about the numerical chromosomal aberration. So, what is numerical aberration? Numerical aberration are those aberrations that involve a change in chromosome number. So, by any means, if there is change in chromosome number of an individual, then that will come under the category of numerical chromosomal aberration. So the organisms are usually, they are usually diploid, and any deviation from this diploid set of chromosome will represent numerical chromosomal aberration, and this will also referred as heteroploidy. Heteroploidy means different. Hetero means different. So ploidy will be different. It will not be 2A. So chromosomal variation is divided into two parts, euploidy and aneuploidy. Now, what is euploidy? So euploidy differed from diploid. Euploidy differed from diploid. What is diploid? Diploid is the 2N standard chromosome complement of a cell. So euploidy will differ from diploid or 2N. That means it may be either N, 3N, 4N, etc. Whereas aneuploidy means it differ from diploid, that is standard chromosome complement. But how? How it will differ? It will differ either it will have one or two less chromosome, or it will have one or two more chromosome. That means either it will be hypoploid or it will be hyperploid. So here in this classification, you can see this ploidy is divided into two parts, euploidy and aneuploidy. Euploidy we have already seen, monoploid, diploid is the standard structure. Then polyploidy. Polyploidy will have two types, autopolyploidy and allopolyploidy. As the name suggests, auto means self. Self polyploidy means the chromosome will be doubled. The same cell, the source of chromosome will be same, and it will get doubled. Whereas allopolyploidy means allo means different. That means the source of chromosome will be different. So we will see in the coming slides. We will discuss more about the autopolyploidy and allopolyploidy. Now what about aneuploidy? So aneuploidy two type, or it is classified into two parts, hypopolyploidy and hyperpolyploidy. So in hypopolyploidy there will be monosomy, nullisomy, whereas in hyperpolyploidy it is trisomy, tetrasomy, etc. So this is the brief classification of ploidy. Now we will see one by one. So before seeing the details of this ploidies, let us see what is the mechanism of euploidy or polyploidy, how this polyploidy is being generated. So first mechanism is failure of pulling apart of two chromatids to opposite end after metaphase stage of mitosis. So in mitosis, when we were studying about cell division, you might be remembering that at the metaphase of mitosis, after metaphase of mitosis, the chromatid of the chromosome, two chromatid of chromosome start getting separated from each other towards the opposite pole with the help of traction fiber, chromatin fiber. So what if they are not getting pulled towards opposite pole? So in that case, there will be doubling of chromosome, and this is being done with the help of colchicine. Colchicine is the agent which will destroy the chromatin fiber or traction fiber, and this may be resulting into the euploidy or polyploidy. Second mechanism is reduplication of chromosome without dissolving of nuclear membrane. So chromosome will be doubled, but nuclear membrane will not be dissolved. So that will also create the euploidy or polyploidy. Then failure of cytoplasmic division. So again in this case the cytoplasm will not get divided, whereas this chromosome will be duplicated. Then multiple fertilization is a rare case, and somatic doubling is the same as somatic doubling means the failure of pulling apart of two chromatid. So these are the mechanism of euploidy or polyploidy, how this euploidy or polyploidy are being created. Now, what is euploidy or polyploidy? So euploidy is of two types. The first one is haploid. Haploid means there will be only one set of chromosome. So individual or somatic cell having only one set of chromosome is called as haploid, or it is also called as monoploid. Whereas polyploid means individual or somatic cell having three or more than three set of chromosome. Because if the individual is having two set of chromosome, it is called as diploid. So individual having two or more than two set of chromosome is called as polyploid. Example, in fish there will be triploidy condition, oyster triploidy condition. In some of the chicken also you will get triploidy condition. Whereas in domestic wheat you can see the hexaploid condition in domestic wheat or modern day wheat. So these are the examples of euploidy or polyploidy. Then polyploids are of two types, the autopolyploid and allopolyploid. Autopolyploid, as I have already discussed, that there will be more than two copies of the same genome. More than two copies of same genome. Suppose the initial one was having only one autosome, A. Then by any means this A is getting repeated and it becomes now 3N. That means the same A is having three copies of A set of chromosome, or the four copies of A set of chromosome. So these are called as autopolyploid. So this autopolyploid, the best example of autopolyploid is wheat. Wheat we have already studied that it is a hexaploid. Hexaploidy means the source of chromosome is the same, that the initial one, 6. Initially the number of chromosome of wheat, they are getting doubled and it becomes 6N. So the polyploidy, autopolyploidy example is wheat. Similarly, allopolyploids. Allo means different. The source of chromosome in that particular condition is different, or it is coming from two different species. So this is here you can see the example 4N, but there will be 2N of A chromosome and 2 set of B chromosome. So this is called as allopolyploid. So this example of autopolyploid, allopolyploid is more, actually autopolyploid is mainly observed in plants or even insects, but in animals the condition is very rare. Similarly, allopolyploids also, it is not common in animals. But if you want to know the specific example of these uh, chromosomal aberration in animals, then it's better that you refer the book veterinary genetics veterinary genetics veterinary genetics by Nicholas. So you go to library and you to try to find out the book veterinary genetics by Nicholas. There in that book you may get the examples of these chromosomal aberration in animals. Now, what is aneuploidy? So aneuploidy we have already discussed. There will be either one or two chromosome less or one or two chromosome more than the diploid chromosome. So changes in the number of single or few chromosome within a set of chromosome. So within a set of chromosome, either there will be one or two less or one or two more chromosome. So example is the monosomic diploid chromosome having one less chromosome, and the formula for this will be 2n minus 1. 2n minus 1. Another one is nullisomy. Nullisomy, here diploid set of chromosome will have one pair of less chromosome. So this is 2n minus 2. 2n minus 2. Then double monosomic. Double monosomic means two non-homologous chromosomes will be missing. So it is important to note that here it is two non-homologous chromosomes. So the formula for this is 2n minus 1 minus 1. That means the two non-homologous chromosomes. Initially, in case of nullisomy, diploid set of chromosome was lacking one chromosome pair, but here there will be two non-homologous chromosomes will be missing. So there is difference in the formula that you have to note. Then the changes in number of chromosome, number of single or few chromosome within a set of chromosome, that means one or two chromosome more. So the first one is trisomic. Diploid having one extra chromosome of a homologous pair, that is 2n plus 1. Then double trisomy. Diploid having two extra non-homologous chromosome. So as it was double, in the previous slide we had seen the double monosomic. Similarly, double monosomic. Similarly, here you can note the this was double monosomic. Similarly, here you can note the double trisomy. Double trisomy. So what will be there? Two extra non-homologous chromosome, and the formula will be 2n plus 1 plus 1. Then tetrasomy, the 4 copies instead of the normal 2 of a particular chromosome. So of a particular chromosome there will be 4 copies instead of 2 normal copy. There will be, that means one pair will get replicated, that is 2n plus the formula will be 2n plus 2. 2n means diploid plus 2 extra chromosome. Now what are the mechanism for creation of aneuploidy? So we have seen the mechanism of creation of polyploidy. Here how these aneuploids are being created. The first one is meiotic non-disjunction. Meiotic non-disjunction. That means the failure of homologous pair of chromosome to separate during meiosis. So there, in case of polyploids, there was failure of separation of chromatids after metaphase. Whereas in this case, it is meiotic non-disjunction, that is failure of homologous pair of chromosome to separate during meiosis. So it will be of two types. Half of the cells they will be carrying n plus 1, means if there will be failure the chromosome will go towards one pole in one kind of cell, and in another kind of cell there will be lack of that chromosome. So half will have complement n plus 1, whereas the other half will have complement n minus 1. So it can occur with any of the autosome or heterosome. It can occur in autosome or in sex chromosome even. Then another is occurrence of translocation. We have already studied about the translocation in the structural chromosomal aberration. So you can take a cue from that. So occurrence of translocation can occur through simple translocation. So this will occur through simple translocation. In human, translocation of chromosome 14 and 21 is very common, and which is known as Down syndrome. So example of Aneuploidy is Down syndrome. So these are the two mechanisms, the meiotic non-disjunction and occurrence of translocation, by which the aneuploids are being created. Now, these are the examples of aneuploids, but these examples are most commonly observed in humans. There are few examples which are observed in animals also, but these are mainly restricted to humans. Here you can see the Down syndrome, where trisomy of 21st chromosome, Edwards syndrome, trisomy of 18 chromosome, then Tau syndrome, trisomy of 13 chromosome, Klinefelter syndrome, this is the trisomy of X chromosome. Then Turner syndrome, there will be lack of X chromosome, lack of one X chromosome. Then XYY syndrome, extra Y chromosome, then triple X syndrome. So these are the syndromes which these are the aneuploids which are commonly observed in humans. You can see what will be the karyotype complement in this slide. Then what are the incidence rate in humans, and that too mostly in the Western population. In Indian population, the data means I could not find the incidence of this abnormality in Indian population, but these all are the incidence in European population or Western population. Then what are the main features? You can note the features of these syndromes, although these all features are of human, and you can refer some standard textbook and you note down the features. You can see the pictures of these individual syndrome in the books. So these are the commonly observed aneuploids in case of humans. So the book that I have told you, the veterinary genetics by Nicholas, there you can find few examples from animals also. So I will recommend you to refer that book for noting down the examples from animal side. Now the commonly observed sex chromosome aneuploids. Commonly observed sex chromosome aneuploids in human. So the monosomy of X chromosome, that means one X chromosome is getting deleted, and the complement now will be X0 instead of XX. So this is observed in pigs, horse, cats, and affect, what will be the effect? There will be ovarian hypoplasia. Then trisomy of X chromosome, that is triple X chromosome, observed in cattle. Again there will be ovarian hypoplasia. In case of male, the extra chromosome, extra X chromosome. When in case of female, if there is extra X chromosome, it is observed in cattle, pig, sheep, dog, cat. And here there will be testicular hypoplasia. When extra chromosome, X chromosome is in male, then it will cause testicular hypoplasia, whereas if it is in female, then it will cause ovarian hypoplasia. Then tetrasomy. Tetrasomy means 2 extra X chromosome. It is observed in horse, and it will cause the intersexuality. So these are the commonly observed sex chromosome aneuploids in case of animals. So I hope I have discussed uh, about the numerical chromosomal aberration, what is needed for a BBSc and AH student. But uh, also I will recommend you to go through the book Veterinary Genetics for getting the examples from animal side. So that will supplement your knowledge, and that will complete the chromosomal aberration. So the chromosomal aberration we studied in two parts: structural chromosomal aberration and numerical chromosomal aberration. So with this chromosomal aberration stands completed. Now in the next lecture we will be discussing something else. Thank you very much.